don't know. Is it the elevator?
I'm also curious.
Yeah, it's always the same sound all the time. Yeah.
Maybe the metro.
It might be the elevator. I hadn't heard it until you asked. I hadn't noted it until you
Yeah,
asked.
but it's omnipresent.
Good morning.
Morning.
Okay, good morning, everybody. Thanks for joining us early in the morning. My name is Siawoosh Mohammadi, and together with Martina Callahan, we organized this symposium on MRG ratio mapping. We believe that it's a good time to have this symposium because the technique MRG ratio mapping has been taken forward by neuroscientists and clinicians. So in recent years, there were many clinical application studies actually, but at the same time also the methods part of this approach has moved forward. And this symposium is about giving the new insights that the field has gained in the last years to share that with you. Now, Martina, who couldn't join us, will give a short summary of the symposium and the talks, and I'm very happy to have the three experts here in their fields who will then give their parts of insights, methodological insights about MRG ratio mapping and myelin mapping, and the function, of course, related with it. Okay, I think the video can start now.
Hello everyone, my name is Martina Callahan. Siawoosh Mohammadi and I have coordinated this symposium on G-ratio mapping as a tool for neuroscience, which will explore the potential of this approach as well as the hurdles we currently face. We hope that you find it useful. In this symposium, we will discuss the MRG ratio, highlighting its advantages, its limitations, and how it can potentially be used in clinical research in neuroscience. We aim to explain how different microstructural tissue compartments influence the MRI signal and how the G ratio can be estimated by isolating the signal from these different compartments. Finally, we will outline the relationship between neuronal conduction velocity and tissue microstructure, in particular focusing on how it is related to the G ratio. The symposium is structured as follows. We will first have 4 presentations, after each of which we will have time for brief questions, but there will be then time for a fuller discussion with audience participation at the end, where we ask you to provide us with questions or discussion points that you would like to see addressed. The first talk will be given by Thomas Nusk from the Max Planck Institute in Leipzig, Germany, who will talk about the neuronal conduction velocity, how it is related to the G ratio, and which other microstructural parameters are relevant for inferring function from microstructure. The second talk, given by Jennifer Campbell from McGill University in Montreal, Canada, will introduce the MRG ratio mapping framework, discuss new methods for MRG ratio mapping, and outline their potential as well as associated hurdles. The third talk, given by Jose Marquez from the Donders Institute at the Radboud University in the Netherlands, will focus on the gradient echo signal and show how the G ratio can directly be estimated from this signal using biophysical models. Finally, in the last talk, Siawoosh Mohammadi from the University Medical Center in Hamburg-Eppendorf, Germany, will talk about the calibration step in volume fraction-based G ratio mapping and where the current problems exist and how they can potentially overcome.
Okay. Yeah,
welcome everybody also from my side. I'm happy that at least some people have found their way out of their beds. So my name is Thomas Knüsche, and I'm the one who will talk not about georatio mapping, but georatio using, actually. So I would like to present. I really have difficulty to see the screen. I think I have to, because that one is flashing my eyes. Can I use a microphone, maybe? So that one, okay, much better. So I would like to talk about axonal transmission and how we can use modeling to relate non-invasively obtained structural information and functional data by means of modeling. So neuronal modeling is concerned with mimicking the function of neurons, of course, which means the temporal evolution of membrane potentials and action potential rates and so on, and of course the transmission of action potentials within certain brain areas where we have short delays which we can often absorb into the dynamics of the cells, but also longer delays which we can't ignore anymore, and of course everything in between. So an axon is bluntly characterized by its length and the transmission time we have there, which adds up to the transmission speed, which is the most characteristic feature of an axon. And in terms of structure, we have properties like the diameter, the axonal diameter, of course, the thickness of the myelin, characterized by the G ratio, and also things like the size of the nodes of Ranvier and their internodal distances. So if you want to model this, if you want to capture that by a model, there are very complex, detailed numerical models around. However, they have the problem that they have many parameters which we have to also to know, which is often not the case, and they are also computationally very demanding. So if you want to model many axons at the same time, you have a problem. So that's why we developed a simplified analytical model, which has two parts. The first one is a passive transmission part, which is simply characterized by the cable equation, as you can see here. And the active input at every node of Ranvier, the restoration of the action potential, can be modeled in a complex way by, for example, the Hodgkin-Huxley formalism. However, it is kind of overkill if we solve these Hodgkin-Huxley equations for each node of Ranvier, knowing that it is always the same thing. So we can use a stereotypical response at every node that is elicited once the membrane potential exceeds a certain threshold. And that one we can compute once, either using a detailed model or also using empirical data. If you want to know more about this kind of approach, I just refer you to this publication that is cited underneath. Here, I would like to show what this model actually can do. So first of all, of course, we want to predict the velocity as a function of the axonal diameter, which is, as we know, the most important parameter. And as we can see here, if we make some reasonable assumption about the length of the internodal length and the G ratio, then we end up within the experimental range, and also we have this almost linear relationship between diameter and velocity without further tweaking the model. So that's already nice. So what about the influence of node and internode length? It looks like this. Yeah, so we also make some assumptions about the other parameters, as you can see here. And we see that the velocity depends on the ratio between node and internode length, optimally about 40. And of course, also the absolute value, the shorter, the faster. And if we make the reasonable experimentally verified assumption that the internodal length is about 100 times the diameter, and assume that the node length maybe varies between 1 and 2 microns, which is also quite royal, then we then we have a very short, very small variation of velocity, actually. So that means node and internode length are maybe secondary parameters. And finally, the myelin thickness, so the topic of this symposium. So we get a relationship like this, yeah? If we map velocity against G ratio, which depends on the diameter, as you can see here. So for larger diameters, the G ratio dependency becomes stronger. And for larger G ratio, that means thinner myelin, the diameter dependency reduces actually. And mathematically, this really qualitatively fits this age-old relationship that has been known already for 70 years now. Although there are some detailed differences, or differences in detail. So this exponent of 0.5 is maybe a bit higher, maybe 0.7 or 0.8, according to our calculations, but qualitatively it is the same as has been known before. So, however, now we have talked about a single axon. But the relevance of single neurons and axons in modeling is limited for several reasons. First of all, of course, it's prohibitively expensive to model any relevant brain network by single neurons and single axons. But there are more reasons. One of them is, of course, that the functional measure that we want to fit to our model to are also that are available if they are non-invasive are sensitive to cumulative activity of many neurons, actually. Also, the structural properties we want to have, or we need to characterize the models, are also mostly available at the population level rather than single axons. And finally, also cooperative action of many neurons seems to be also relevant for function, although that is a controversial issue. So it's not quite clear so how much a single neuron matters and the population activity matters. So that means we have to, or we want to model axon bundles, yeah? So we have many axons that run in parallel. We can find the direction, for example, by tractography. We might have some distribution that we can measure of diameters, as we can see here. So in the end, our parameters that we deal with are distributions of axon diameter, G ratio, node length, and so on. So of course, we could now say, okay, we take models of many axons of different diameters and so add them up, and then we have the behavior of the bundle. However, there is something in addition to that. Rather than just adding up the behavior of all the single axons, we have mutual influence, or we might have mutual influence between axons, so-called apoptic coupling. So if you have a bundle, and one of these axons has an action potential, then we would measure an extracellular potential in between the axons, which causes a perturbation potential in other axons. However, if you look at the magnitude, it's very small, so it shouldn't influence much other axons. In contrast, experiments already in the 40s have shown that there is a apoptic coupling between axons, which can synchronize action potentials, slow the transmission down, and even elicit action potentials in passive axons. However, these were quite artificial situations, so the axons were ex vivo embedded in highly resistive media. So how would that look in a more realistic situation? So if you now consider one axon, this is this black dot there, surrounded by many other axons, yeah, that carry action potentials. Then, of course, the first thing which is immediately clear is that all the effects will add up. So if we first assume that all these action potentials and these surrounding axons are perfectly synchronous, so they are at the same time, at the same place, then if we look down, yeah, if you increase the size of this axon bundle, then the effect, the extracellular potential, will reach quite some magnitudes. So it has a spatial profile of an action potential. It's similar to an action potential, and the magnitude will amount to several hundred millivolts, which is, of course, way enough to influence other axons. And this effect is already present if we have quite small bundles. However, in reality, of course, all these action potentials wouldn't be synchronous. Yeah, they would be somehow distributed. So if you look at this situation, for example, might be distributed over a length of 2.5 millimeters, 10 millimeters, or even 5 centimeters. Yeah, then you can see that the extracellular potential goes down to, for example, if it's 50 millimeters, several millivolts, which is still something that could influence firing in, or transmission in other axons. So if you take this last situation and assume realistic diameters like half a micron or 1 micron corresponding to 2 or 4 meters per second transmission speed approximately, then this means we are looking at firing rates of 40 or 80 hertz, which is something that realistically occurs. So that might be a realistic case. So let's look at this case further and see how the different parameters of the axon bundle, how the different structural parameters influence the effect that we see. What kind of effect do we see? If you look at the mean axonal delay, then we see that this axonal delay is decreasing. That means the speed is increasing. And this depends on several parameters. Of course, it depends on the intensity of the volley I put into this axon bundle. So what percentage of all the axons is actually carrying an action potential. And we see that this intensity, the higher the intensity, the lower the delay, that means the higher the speed. The second parameter is the duration of the volley. The shorter the volley, the more synchronized all these action potentials are. If they are more synchronized, we have more speed up. And finally, also the bundle diameter plays a role. The thicker the bundle, the more apoptic coupling, although this converges after some time. So these are the influencing parameters. So what can we say? We can say apoptic coupling in heterogeneous fiber bundles accelerates action potentials, actually. And this acceleration increases with input intensity, synchronization, and bundle diameter. However, as I've already mentioned, previous experimental and computational studies have found the opposite, a deceleration and also resynchronization due to apoptic coupling. However, in these situations, different conditions were assumed. For the computational studies, it's mostly a small number of identical axons that have been looked at so far. And in the experiments I already have mentioned before, isolated axons that were embedded in highly resistive material were looked at. And if we mimic these conditions in our model, we also can reproduce these results, with a deceleration and synchronization. So that puts further credit to the model. So just let me summarize and conclude. So we have a simple and accurate model of action potential transmission through fiber bundles. The most important parameters that determine that model are G ratio and axon diameter. So these we would like to determine individually. Other parameters might be taken from the literature just approximately, like node length and so on. Axon bundles have properties that are beyond the sum of axons. So this is done happens through apoptic coupling. The effects of apoptic coupling depend on functions, so the intensity and the synchronization of spike volleys, and also on structural parameters. And that is what we actually want to have from our MRI people. So we want to have individually determined geometry of axon bundles, of course, and the microstructural parameters, most importantly the G ratio and the diameter. And with that, I would like to thank you again for your coming and attention, and also my collaborators, most of all Helmut Schmidt, who did all this modeling work. Thank you very much.
Thank you. Are there any questions for Dr. Knizia? You'll have another chance at the end, but don't be shy. I'll pass you the microphone.
So haptic coupling happens in many neurological diseases, so notably MS, but you also see evidence of it in others. Do you think that with your model you can estimate the divergence between normal transmission and haptic transmission, giving you a surrogate that can give a measure of neuronal dysfunction and degeneration?
So you mean that I can project from what I observe in terms of haptic coupling?
Yeah, so your model... Yeah, so your model will estimate what the idealized situation and then the divergence between your model and what you measure in vivo gives you a measure of the haptic coupling that's happening.
So, and the question is then...
do you think that would work?
So in terms of, so I could fit the model to that, and I could project this observation to structural changes that I would expect, for example, demyelination or, I don't know, thinning out of the bundles or bigger distances. Yes, That would be inverse fitting of the model. That's something we would like to do. So if we would have such data.
Yeah, that would be really cool.
Yeah.
Okay, we'll move on to the next talk now, but we'll have a chance to discuss at the end. So the next talk will be by myself. I'm Jennifer Campbell, and I will talk about the aggregate G-ratio weighted imaging framework and recent attempts to go beyond it. So I'll spend the bulk of the talk talking about tissue models and the accompanying imaging frameworks that have been used to estimate the G-ratio from magnetic resonance imaging. I'll start with what we call the volume fraction-based framework, and then talk about alternatives to this framework. I'll then talk about future directions to try to go beyond a single G-ratio estimate per voxel, with a focus on estimating a G-ratio for each of multiple fiber populations within a voxel. Really, the whole talk will be about challenges for doing this, but I will summarize the challenges at the end. So here we have a scanning electron micrograph of white matter. And now it has been segmented into 3 compartments: the interaxonal compartment shown in blue, the myelin sheath shown in red, and everything else, which we'll call the extraaxonal space. So we can create a simple tissue model from this. And the G-ratio, as we've seen already, is the ratio of the inner to the outer radius of the myelin sheath. So as Thomas Kanija told us in the previous talk, the G-ratio is a key determinant of conduction velocity and hence of function. And there have been a couple studies to date using MR estimates of the G-ratio in the context of conduction velocity. There have also been a number of studies in healthy white matter, including in development and aging, and also in disease, for example, multiple sclerosis. So just to motivate why we want to measure this parameter, in the example of multiple sclerosis, traditionally we look at myelin-sensitive metrics with MRI, but we can have a reduction in the total myelin for multiple reasons. So in the middle frame here, you see we could have minor axon loss with the remaining axons experiencing demyelination and incomplete remyelination. Or on the right, you see that you could have more extensive axon loss with the remaining axons having a relatively healthy G-ratio. So really the key point to estimating the G-ratio is to be able to decouple the relative myelin thickness, which is the G-ratio, from the total fiber volume. So now I'll describe the volume fraction-based framework for estimating the G-ratio. Here we have our simple tissue model again. And we have the myelin volume fraction, the axon volume fraction, and the sum of these two is the fiber volume fraction. And it was shown by Nicholas Stikov that an aggregate G-ratio in the voxel can be estimated from any 2 of these 3 parameters without explicitly calculating any diameters. So here you see the G-ratio expressed as a function of the myelin volume fraction and the axon volume fraction. And it's determined entirely by the ratio of these 2. Alternatively, we can express it as a function of the myelin volume fraction and the fiber volume fraction. Again, it's determined entirely by their ratio. So if we want to estimate a G-ratio weighted image, one option is to first estimate volume fraction weighted images and calculate the G-ratio from those. So let's start with the myelin volume fraction. The myelin compartment consists of both myelin water and macromolecules. And there are many MRI contrasts that are sensitive to the total myelin. And if we assume our simple tissue model is correct and that the myelin marker is specific to myelin, which it probably is not, let's call the myelin marker M. We can assume that the myelin volume fraction is an appropriately scaled version of our myelin marker. So Siawoosh Mohammadi at the end of this symposium will talk about what happens when this very simple relationship is inevitably not correct. But for the time being, we'll assume we can estimate a myelin volume fraction. So now we can put that myelin volume fraction into our G-ratio formulation like so. So now how do we get the fiber volume fraction? we have a couple of options with MRI. One is diffusion imaging, because the diffusion displacement distributions in the different compartments will be distinct. Another possibility that we'll come back to later is gradient echo images, where, for example, the phase of the different compartments will be distinct. So in Nicholas Dickov's early work, he looked at simulations of diffusion in straight parallel fibers and determined that there's a quadratic relationship between the fractional anisotropy of the diffusion tensor and the fiber volume fraction. So he took this simulation-based equation and put it directly into the G-ratio framework. Now, the fractional anisotropy will be expected to be acquisition dependent and also extremely dependent on fiber geometry. So we asked whether we could get out of this straight parallel fiber regime. And to do so, we asked whether we could estimate the axon volume fraction from more complicated diffusion models that take geometry into account. Now, a note that in a typical diffusion acquisition, we're operating at a relatively long TE. And what I'm showing here is the T2 decay curves of different compartments. Both the myelin water and the myelin macromolecules have short T2 values and will have negligible signal at the TE used in a diffusion acquisition. So the signal fractions we're getting from a diffusion acquisition are fractions of the remaining spaces. So for example, we can get what I'll call the intraneurite signal fraction. So what we did in our original formulation was we took the intraneurite signal fraction directly and put it into the G-ratio formulation, treating it as a volume fraction. So to compute the total axon volume fraction, we need to incorporate our already estimated myelin volume fraction. And that gives us an axon volume fraction shown here, which we can put into the G-ratio formulation like so. Now, there's a problem with this. Recent work in the last few years using multi-TE diffusion acquisitions has shown that, in fact, the T2s of the intra-axonal and extra-axonal spaces are different. And this difference might be regionally variable, and in pathology, it can be quite a bit larger than in healthy tissue, and the polarity reverses. So we really need to take T2 of the compartments into account in order to go from signal fractions to volume fractions in this very geometrical framework. So just to spell out what's happening here, if you have equal volume fractions of the intra-axonal and extra-axonal spaces shown here, what you're measuring at the longer TE is a signal fraction, so they will no longer be the same. And if you assume no spatial variability in the T2s of these spaces, despite the fact that they're different, then the mapping from the signal fraction to the volume fraction will be a scaling. If there is spatial variability, then this mapping will not be a scaling, and you can end up in a scenario where a certain region has a truly elevated G-ratio compared to another region, but it appears to be decreased. That's because the difference in the volume fractions is changing. So we need to take T2 into account. So here I've introduced a parameter F0, which is the signal fraction corrected for relaxation. And if that's corrected, ideally by measuring the T2, then the G-ratio formulation becomes this. So now I'm going to add a compartment to our tissue model. We may have free water that is far away from the other tissue structures. And this free water will also have different relaxation parameters and a different diffusion displacement distribution. Its signal fraction can be estimated from diffusion MRI again. And we can then calculate the axon volume fraction, incorporating it, and put it into the G-ratio framework, noting that T1 might become relevant in such a long T1 compartment. So now, we also might have a volume in the extra-axonal space that is diffusion invisible, that is not myelin. And in fact, when we adjusted the axon volume fraction to take into account the myelin, we should have taken into account this extra extracellular volume fraction that is not myelin as well. And once we are using both of these, I'm not certain how to measure and differentiate them with MRI. This is an outstanding challenge in the field still. But if we can, then we put them into the G ratio formula like so. But we also need to consider the intraaxonal volume that is not myelin. So we could also reformulate this as a correction to all diffusion model-based signal fractions where we need to correct for both relaxation and diffusion invisible macromolecular volume. Excuse me. So now we have this formula for the G ratio. So there are other concerns that I'll summarize at the end, but I'm going to move on to alternate frameworks for estimating the G ratio. There is a model called the hollow cylinder fiber model that expresses the myelin-induced frequency offsets of the myelin extraaxonal and intraaxonal spaces directly as a function of the G ratio. And so we can compute the G ratio directly from the complex GRE signal, as has been done by Mark Drake-Smith here. Taking this a step further, excuse me for a moment. Mikhail Kartar has done a similar approach with the hollow cylinder model, and I'll come back to this in a little bit. Taking it another step further, Jose Marques will talk about this at the end of the symposium, but it's been shown that the signal is better described by more realistic axon geometries, and he has used a machine learning framework to dissociate the fiber volume fraction from the G ratio using the GRE signal again. Another way to look at this, which you might have thought of when we talked about the dependence of the fractional anisotropy of the diffusion tensor on the fiber volume fraction, is that the extraaxonal diffusion anisotropy, this d delta shown on the left, is dependent on the amount of myelination, even though we don't get direct signal from myelin in a diffusion acquisition. And so it can be expressed as a function of the G ratio and the diffusion model parameters. This Fs is our relaxation-corrected intraneurite signal fraction that we talked about before. So the G ratio can be computed from the diffusion signal alone, and this was proposed by Marcus Nielsen and Bjorn Lampinen. So now I'll look at future directions in G ratio mapping. It would be nice to estimate a full G ratio distribution in the voxel. But what I see as a step toward that is getting a G ratio for each of multiple fiber populations. So in fact, the approach that I whirled past by Mikhail Kartar is a diffusion prepared phase sensitive acquisition that allows for the estimation of a G ratio for multiple fiber populations in a voxel. This is work in progress and requires rotation of the head in the scanner, but it is an intriguing idea. He has now taken the relevant fibers G ratio and mapped it along the tracks of the brain. If we go back to the volume fraction-based approach, if we want to estimate multiple G ratios in a volume, we need to estimate multiple volume fractions in a voxel. And I'll start with the myelin volume fraction. There's been quite a few recent papers looking at estimating relaxometry parameters, such as T1, along each of multiple crossing fibers in a voxel, shown here on the right. There's also an approach called commit that allows for the estimation of really any quantitative MRI parameter along a streamline or bundle, assuming that it is constant along that streamline or bundle. So here we have bundle-specific myelin water fraction, bundle-specific magnetization transfer saturation, and bundle-specific magnetization transfer ratio. So what Simona Schiavi has proposed is to also use commit to estimate an interaxonal volume fraction and therefore estimate a streamline-specific G ratio. So now I'll summarize the challenges that we've seen. we need to take into account relaxation in order to estimate volume fractions for the G-ratio framework. We also need to take into account, in diffusion modeling, the non-myelin diffusion invisible macromolecular volume. It would be nice to estimate a G-ratio distribution and not just an aggregate in the voxel. Siawoosh Mohammadi at the end of this symposium will talk more about specificity and accuracy of myelin markers. And diffusion modeling does have other issues that I didn't go into, such as degeneracy of the models and incorporation of exchange. We didn't talk about applicability in gray matter and validation of these techniques, but perhaps we can go into more detail in the panel discussion. And with that, I'll conclude that G-ratio weighted imaging is a way to interpret MRI data that are sensitive to both myelination and axonal volume. But G-ratio weighted imaging is limited by the specificity and accuracy of these metrics. And we have recent methodological advances that may aid in estimating more than one G-ratio per voxel. So thank you to all of my collaborators and to all of you here at this symposium. And I'm happy to take any questions now.
Good morning. This is really interesting. I have a question about validation. So I wonder if there's any ground truth validation either in a test tube or in postmortem tissue to any of these models.
Yeah, so I cut that out of my talk in the interest of time. Catherine West and Mark Does have done some pretty extensive validation in ex vivo mouse models. So you can look up their work. It's nice in that they had multiple knockout models, so they have hypermyelination and hypomyelination, and they were able to look at G-ratio using a number of different diffusion models and myelin markers. Just a note that you can either validate this, if you're using the volume fraction-based approach, you can potentially validate this by looking at the volume fractions directly or at the G-ratio. But it's difficult to do it with the volume fractions because the axon volume fraction requires the myelin volume fraction to get it quantitatively correct, right?
Maybe time for one more question.
How much you can just interpret this type of G ratio metric at the voxel have some partial volume with a gray matter, because maybe the modeling assumption doesn't want the gray matter, but some parts of white matter interface is a gray matter. I'm just wondering how to handle partial voluming.
Yeah, so I think it's an ongoing question to what extent this holds in gray matter. If this model held in gray matter, it wouldn't matter if your voxel was half gray matter, half white matter, but I think it's an open question what diffusion models mean in the presence of dendrites, and you'll probably have more non-myelin macromolecular content. So open question, be careful about what you're doing and make sure you know what you're looking at.
Thank you. So, good morning. My name is Jose Marquez, and I'm from the Donders Institute at Radboud University. And now I'll be giving a presentation on how can we try to use generative biophysical models that use the most simple MR signal that there is to try to decode and squeeze out G-ratio information out of that signal so that we can have G-ratio maps throughout the brain. So, but before we can go into the detail of how could we try to measure this ratio between inner and outer diameter of outer radius of an axon, it's actually important to understand how does this black stuff, which is around the axon, is affecting the MR signal, because that is what will give us access and open a window towards be able to measuring G-ratio. So as we already saw before, myelin is this phospholipid layer which is surrounding the axon. And well, phospholipids is not something that we can see, but luckily, in between these bilayer phospholipids, you have a bit of water. And so if we simplify the world, like physicists like to do, we can say that, well, brain is constituted by 3 compartments. There's a free water compartment. There's this small water that is kind of trapped within the myelin layers. And then there's myelin lipids. And this is actually not very far away from true. Let's say our brains are mostly water, and if you take all the water, 80% of your weight is actually myelin lipids. Now, what's interesting, well, we can't really see very well myelin directly, which would be really useful unless you use ultra-short acquisitions. But the way we try to often look at how much myelin there is in the brain is by using the fact that it has a short T2, but not so short that we can see it. And so we typically acquire these multi-echo data sets, and we separate the very short T2 component, this blue compartment, and the long T2 compartment. And then we say, well, the myelin water fraction must be the ratio between these 2 compartments, the one that decays very fast and the way that decays very slowly. Other people have also started noticing that actually this is not a property only about T2, but you also have an apparent T1, which is different in this myelin water compartment and the free water compartment. And because there's so much lipid out of it, and there's so much exchange, and there's various types of exchanges, chemical exchange between the free water and the bound water, and you also have all sorts of magnetization transfer. Actually, everything that you measure with MRI is usually quite sensitive to these lipid, the lipids in myelin. So, but these are really kind of very complicated processes to model. So I don't like them, and I like to focus on this hollow infinite cylinder model, which is another way that we try to simplify how the world is. And for physicists, if we don't have just 3 compartments, now we have 3 other compartments, and we just have white matter is just one long cylinder, where we have the intraaxonal water, the myelin compartment, the extraaxonal water. And as we know, we have these lipids here, these lipid bilayer, and these lipid bilayer is constituted by these very long, elongated macromolecules. And what's interesting with the elongated structures is that they tend to have an anisotropic susceptibility. And then anisotropic susceptibility is a very complicated thing, but it just means that once you put it inside a magnetic field, it will be magnetized, and it will perturb the magnetic field. And because you have so much myelin, and it's so well structurally organized around the axons, it perturbs the field in a very organized way. So if you look here, this is an example of a 3D EM, and you see that somehow, as soon as you introduced these magnetic materials inside the scanner, and this is with a bundle of axons which is perpendicular to the magnetic field, suddenly you have a different color of the intraaxonal compartment versus the extraaxonal compartment. So the intraaxonal compartment is darker. And this is really kind of what starts giving us a chance to use gradient echo signal to see G ratio, because it's starting to separate this free water compartment into an axonal compartment and an extraaxonal compartment. The sad thing is that if you have these axons and they are suddenly parallel to the magnetic field, actually there's no difference whatsoever between the inner and outer compartment. But luckily you still have a bit of a difference in this myelin water compartment. I'll try to now show some more simplistic examples of what this means, what these perturbations of the magnetic field mean in terms of MRI signal. So let's consider these even more simpler white matter models. Let's consider one axon which has a given FVF, or two white matter models with the same FVF but different G ratio, so thin myelin sheet and a very thick myelin sheet, but the same FVF. And another one which has a much higher fiber volume fraction, so the fiber occupies a bigger fraction of the pixel, but it has also a very high G ratio. What would be the signal if you'd put these things inside a magnet? And what you see in terms of the gradient echo signal is that actually you can separate very well this red model, which has this model here, from the other two on the magnet. But you can't really separate these two models that have different G ratios, at least not on the magnitude data. But on the complex data, you actually suddenly have a bit of a hope of separating them, similar to what we had seen on the previous slide. And this is also true when your axons are parallel to the magnetic field, although there it becomes increasingly difficult, as it has been suggested on the previous slide. So it's difficult, but it's not impossible, and that's how we typically do myelin water imaging. But and the reason why we typically do myelin water imaging more than G ratio, and it's that's a more robust method to look into myelination, is because actually what you can see from all these plots is the red is very much distinguishable from the green and pink, and this is actually models which have a different myelin volume fraction. These two have very similar myelin volume fractions, and so they are very similar in the gradient echo signal. So how do we do this in myelin water imaging? Typically what we do is that we get the gradient echo signal and we say, well, there's 3 compartments. There's the myelin water compartment, there's the intra-axonal compartment, and the extra-axonal compartment, and each of them has given a signal amplitude, has given a T2 star decay, and has a different given a frequency shift. So this means that we are actually just getting one signal decay, and we are fitting 9 parameters to it. And that's, I think you need around 5 parameters to fit an elephant. So here we're using 9. We could fit many things. But surprisingly enough, this is actually an extremely robust way of getting myelin water imaging. And you can actually improve on it. This is work by Pokxing in my lab, where he noticed that, well, you can get myelin water imaging using this method, but if you use some of the information that I shared with you on that very busy slide at the start, that myelin water has a short T2 star, but also has a short T1, you can design acquisitions that make your myelin water maps now a lot less noisy. And if you, on the top, add diffusion information about what is the fiber orientation and what is the inter versus extra-axonal ratio by using advanced diffusion models, you can get even higher SNR myelin water fraction. But this is myelin water fraction. And so myelin water fraction is nothing but the amount of water which is trapped between the myelin layers and all the water that you have, axonal myelin water and extra-axonal water. G ratio is something different. G ratio is about volumes, actually. You can see it as a ratio of the radius, but you can also see it as a ratio of the axonal volume versus the axonal plus myelin volume. That's another way of expressing the G ratio. So you can try to do some kind of calibrations about assumptions on what concentration per compartment, and you can convert the measures that you have from myelin water imaging into G ratio maps. But you really kind of have the question of, it is based on assumptions, and will these assumptions actually hold in when you get to pathology, where your myelin might not be as tightly fitted around the axon, and that's something that happens, for example, in MS. So the question that we had was, maybe myelin water is a too indirect way of measuring microstructure, because what we actually want to measure, as mentioned by Thomas, is the fiber volume fraction and the G ratio. And I'd also like to say that we also want to measure how much myelin is actually there, because these two models, they have the same G ratio, they have the same fiber volume fraction, but they have a very different amount of myelin, and this can be showing pathology. And all these metrics here, they actually impact the GRE signal as we saw it. But what the group from Carl Miller has shown very elegantly is that you actually, if you keep considering this very simplified world of cylinders, that white matter are just cylinders, you actually have a very biased view of what white matter looks like. So here we have a packed white matter model based on cylinders, and here you have a real electron microscopy data set, and you see that these two images, they actually are very different, and they not only are very different, they will generate very different MR signals, or they will predict very different MR signals. So ideally, we don't want to use circular models, we want to use electron microscopy models. So ideally, if we want to be able to fit, we would like to be able to have electron microscopy models for all sorts of G ratios, all sorts of FEFs, and all sorts of myelin concentrations. And that's actually impossible. So that's what we try to do. This is work from Renault. I'll try to go through how we approach this problem. It has basically three important steps. One of them is how do we create realistic white matter models? And how do we simulate the kind of signal that we expect? What kind of acquisition we have to acquire, and then how do we train our networks to be able to extract the G ratio maps. So if you can click on the movies to start. So what we started by doing is to go to a database of myelin axons and put them randomly inside a matrix and let them compact. And then we remove some accents so that we can get basically any fiber volume fraction that we want. And we can also change the G ratio by now a posteriori removing some of the myelin. So if you click on the last one, we'll get a bit of a thinner G ratio. And by these, in this approach, we can actually now create a huge dictionary of possible white matter representations that are at least a lot more realistic than the infinite cylinder models. And having these now, it means that we can actually have different white matter models with different FVFs, different G ratios, and we can simulate what's the field perturbation, and we can also go on to simulate their signal. And that's all good, and we can also not only simulate the signal for this one model, we can also then simulate the signal on how does the signal depend as we vary this realistic white matter model in respect to the magnetic field. So the MR signal will be very different depending whether the fiber is running along the magnet, so when you're lying down the cortical spinal tract, or optical radiation, which is going to be always perpendicular. What is interesting here, and what I show here with the same color, is that the MR signal depends heavily on the fiber volume fraction, which have here the different colors. But within each of these groups, you have different white matter models, different of these representations of models which have the same FVF, they have the same G ratio, but they generate only slightly different signals. So it means that we can have a bit of a representation of what is the variance that we expect with realistic white matter models in terms of MR signal. Now, now the problem is, how can we use these dictionaries? We know what white matter models, what kind of signal they generate. How can we use these to extract information from data, from actual data? So we did this ex vivo because ex vivo brains, they are a lot more compliant in terms of rotation. So what we did was that we acquired some DTI and we acquired gradient echo images with multi-echo acquisition. And what we did is that we rotated the brain a lot inside the magnetic field. So we acquired one position where the person, the brain, was standing in the normal supine position, and then we rotated heavily. And now what you have to think is that each fiber that has a given orientation at the start, as you rotate the brain, it will have a unique trajectory of new orientations. So you have to basically go back to your previous dictionary, reshuffle it, knowing how you rotated your brain, and create a new dictionary. And that's how you train the network. So you train a network. This is just a very simple, fully connected network where you try to decode, based on your real and imaginary dictionaries of signal, of what would be the signal for the different orientations. You try to decode what is the FVF, the G ratio, and T2's relative weight of the signal inside the myelin water compartment, and the susceptibility of myelin. So you can think that this is machine learning is often like a black box that is doing magic, but actually all we're doing is to learn how to fit these parameters from data. And we train the network based on our simulations, and then we will fit it on the data that we acquired with the different rotations of the brain. And actually, we're doing a lot less demanding things than we're doing with myelin water imaging. So this is just kind of a sanity check to see if our models are converging and how many rotations of the brain we need to be able to get robust G ratio maps. And the answer is pretty much you need to have at least 3 large rotations to get it right. So you need to at least once get your angle of your fiber well far away from being parallel to the magnetic field. And if you think about it, instead of myelin water imaging, you're doing fitting 9 parameters to one single decay rate. And now on these generative realistic white matter models, we're actually only fitting 6 parameters. So we are actually, with all this complexity, we are actually simplifying our fitting. We're now fitting a very complex numerical model, problem. And this is what we get. So if you for the whole brain maps, which are decoded from 9 head positions, so 9 rotations of the brain, we get FVFs that have some sensible characteristics and some less sensible. For example, the fact that In deep gray matter regions, this tends to fail, but in white matter and gray matter seems to hold quite well. The same thing for the G ratio and the remaining parameters. And what's interesting also is that we actually repeated this experiment with multiple flip angles. So we changed our acquisition so that we would be very sensitive to relaxation parameters. And what we observe is that actually, independently of the amount of T1 weighting, we always got the same values for this G ratio. And the standard deviation across all these measures, it was extremely small. And also quite cool is that you can go from 9 head orientations to 3 head orientations and still get very comparable FEF and G ratio maps. So I think to some extent what the question is, have these generative models, are they a new window for G ratio imaging? Well, I think yes and no. At least we have a framework which only requires on acquiring GRE data, which is the simplest acquisition that everybody can do, and only having 3 head orientations. So that's one positive thing, and it really explores the non-exponential nature of the decay of the individual compartments, the intra-axonal, the myelin water, and the extra-axonal compartment. And so in that sense, it's quite an elegant framework. And it allows us to get G ratio maps which don't have this relaxation weighting dependence that Jennifer was mentioning earlier. And it really separates the G ratio from myelin sheath swelling, which is an important aspect I think in diseases like MS, where this would be an issue. But there are some limitations, and I think the biggest limitation is you actually need to acquire 3 orientations in respect to the V0. And you typically don't ask your subjects to, please now, can you rotate your head preferably 90 degrees before the next acquisition. And it's a pretty complex model that we are fitting, at least in terms of computation. And it still might not be complex enough. Because we are ignoring like a couple of elephants in the room. We are ignoring iron deposition. Is it the same in the intra and extra-axonal compartments? We are ignoring the fact that blood is usually organized along the same orientation as fibers. We are ignoring diffusion, although for a good reason. And so far, we haven't integrated exchange and longitudinal relaxation, which would be, I think, quite interesting. Our future work actually is going to be to try to mix these realistic models with myelin water imaging. And we will still be doing some validation, hopefully, in fresh tissues. We've done the validations in formalin fixed, but formalin fixed and in vivo are very different animals. So we'll see. I'd like just to finish with acknowledgements. Kwok-Shin and Renaud that did a lot of this work, and a lot of more collaborators at the Donders, and you for your attention, and I guess also the nice letter type from Glasgow for those that might have recognized. Thank you.
Okay, I think we don't have time for questions because the time is almost over. Maybe at the end, if you have a question, just spare it for then, and then you can choose who you will ask. Okay, maybe I can start. Yeah, perfect already. I will talk about the calibration step in the volume-based approach, and also give directions how it can be improved. For those of you who are sitting on this side, just be aware that I'm pointing on this slide. I hope you can see it. Okay. My talk is structured as follows. Okay. I start with the calibration that is typically done, which is the scaling of the myelin volume fraction, so first point. And I will show that that's not enough in a simulation study. Then in the second part, I will talk about potential scaling also of diffusion-based volume fractions. And finally, if time permits, I will talk about how that might be used to actually improve our G ratio estimates, and end with summary. Okay, so first time. The simulation is structured as follows. We have basically histologically based myelin volume fraction. We have we relate that in an analytical way to an axonal volume fraction here. And we then, for that, we use histological data. With these two, we can then calculate a ground truth G ratio. And this is shown here. On the other hand, we have simulation-based MRI myelin volume fractions, and they are deliberately chosen that we have one with a large offset, one with a small offset, and different slopes, because these are the issues we want to calibrate out and show how that can be a problem. And from them, we calculate, again, using our MRG ratio formula, a MRG ratio now, and compare this MRG ratio, different colors are the different modeled MRG ratios, to the ground truth one. To do that, we use a data set that Jennifer mentioned already. This is from Mark Doe's data set with the 6 healthy control mice and 3 different myelin models. Hypermyelinated model, P10, with more myelin, and 2 hypomyelinated models. The TSC2 model has almost no myelin. And in each mouse, we now acquire multimodal data. So MRI and histology, electron microscopy, in 4 different regions, 3 in the corpus callosum and 1 in the anterior commissura. Now we can basically calculate these parameters I showed here. To do the first part here, the ground truth axon volume and myelin volume part, we used here all the data available and fitted a heuristic line here between myelin volume, histological myelin volume now, and axon volume fraction. Be aware that here at the end it produces negative axon volumes. You will see what that causes later in the simulation. And from that we calculate also a ground truth G ratio here. Okay. Now we want to simulate also different MRI-based myelin parameters. So we basically have 3 different myelin measures available, and I will go through them now. And this is a figure from our G ratio review, recent G ratio value, with all the different compartments in a voxel that we typically model with it for these different models. The first one would be the myelin water fraction approach, which Jose talked about, and that actually has a quite small offset and a good slope here. The second approach is a bound pool fraction, which has a large offset as compared to its slope, but also correlates very nicely. And the third one is basically the proton density, or also known as the macromolecular tissue fraction, which is 1 minus the proton density. And so you would take 1 minus this slope here to get correlate of your myelin volume fraction. And these 3 we use then for our simulations to generate these model G ratios. Here in a scatter plot, we compared the ground truth G ratio now to the MRI G ratio, and what we found is that bound pool fraction shows almost no reasonable relation in our simulations. So if this is a line of unity, we have in green then the myelin water fraction based G ratio, which shows a reasonable relation, and we have the MTV, the 1 minus proton density approach, which has the ceiling effect, which comes because for very low myelin volume fractions, we got this negative values of AVF. And here is basically the Blunt-Altman plot, and the dashed line shows the error, where the solid line shows the mean. Now, when we do the calibration, what we typically assume with a scaling-based calibration is that there is no offset. So that's the same as if we basically moved out this MRI-based myelin volume out of this equation, and then this term here would be our scaling. And you see it's not really the scaling, it's only the scaling if this beta here is negligible as compared to the alpha. And since in our simulations now we have different relations between the slope alpha and the offset beta, we can look how that will affect our results. So now we basically added the calibration and look at the results. We see that for the bound pool fraction, still there is no 1:1 correspondence. The myelin volume and the MTV-based G ratios change a bit in shape, but when we look at the error, we see that the error actually increases. The only thing that improves is the mean, so the offset, so the bias gets better for all three of them, which makes sense because we basically move the line, the middle line, towards 0 with this calibration approach. Okay, second point is now diffusion. Jennifer nicely introduced the relation between axon volume fraction and what we measure from diffusion, the axon water fraction, that it has to be scaled with 1 minus myelin volume fraction. That's what typically is assumed is true, but we know that basically there is also unmyelinated axons, and when relating the MRI signal to to our model, then we might have to account for an offset to model that correctly. The other thing that Jennifer mentioned was the compartmental T2. And we showed here in this preprint that it can be approximated in the first order as a scaling factor here to account for basically compartmental T2 on a global level in your data. And finally, the last model we will consider is, again, a linear thing where you have scaling and offset for diffusion data as well. Again, we use this nice multimodal data set from the Mark Dose lab. And we now look at the difference between histologically estimated axon volume fraction and the MRI-based. And this is the residual sum of squares that we will look at and calculate the Bayesian information criteria to see which of the models is best. And the number of model parameters correspond here to the calibration parameters. So 0 means no calibration, and then linear model would have 2 calibrations. And the negative the BIC, the better. We, in the preprint, looked at 4 different models. For time concern, I will only consider WMTI Watson, the one model with 5 different diffusion parameters. Here are the results. So we see that BIC is best when we have the linear scaling as expected, but also that when only looking at an offset, we are not too bad. So if we have to choose, so if we don't have enough degrees of freedom, we should just take an offset. And here is how basically the scatter plot looks like if you don't calibrate and if you basically do the calibration with linear calibration. How can we improve the calibration? So the models we looked at here is the 3 parameter calibration, where we have basically slope and offset for myelin, and we have an offset for the diffusion-based measure. Diffusion-based measure was calculated with WMTI, and here we used the myelin water fraction for myelin. And we compared basically the 3 calibration parameter model to a single parameter calibration model, and to make a long story short, we see that the AIC is smaller here, but also that the the scatter plot gets better, Blunt-Altman gets better with 3-point calibration, and that when we basically use different models, different mice for calculating the calibrations, the standard deviation of the parameters are comparable. Okay, to sum up, we have seen that we need an offset for myelin water imaging approaches as well. It is good to take also an offset for the diffusion approaches, at least for the models we have looked at, and that having more calibration parameters in our estimation would help. Future work now needs to find a good method to estimate actually 3 calibration parameters from data available, or to find better diffusion and myelin proxies for the volume fraction approach. One has been recently suggested by Rowley et al. Using the inhomogeneous transfer imaging proxy for myelin. So, and with this I'm done, and we have 5 minutes for questions. Okay, I would also like to acknowledge, of course, Mark Doss first for the data and my collaborators.
We have some time for questions. There was one question on the app about how do we calibrate head orientation, and actually, so what we do is that we do essentially motion correction, co-registration, like you usually do in fMRI, or and then we basically, it means that for each acquisition, given the orientation that the rotations that you might do with your head, then we have to retrain our network that will fit the G ratios. So it's measurement specific.
If you have general questions to all the talks, please feel free to ask now. We have still a few minutes left in the session. Or comments to MRG ratio mapping in general would also be appreciated. Yeah.
Well, maybe this is kind of general, but I think for histological validation, ex vivo neuroimaging can be potentially very important. But just wondering, because ex vivo imaging has its own limitation, like maybe relaxation time, diffusion parameter itself depends upon maybe postmortem preparation time or whatever, something non-biological. Then maybe how to just go beyond this type of issue may potentially impact your G ratio estimations.
Well, our approach was, we know it will be different, but we are fitting all those parameters. So that we'll still get the G ratio that you'll measure ex vivo by having the tissue fixed to some extent. Because yes, it's true the relaxation parameters will change, but we are fitting them. So they are another parameter of our model. Obviously, the fresh water, not the fresh water, but fresh tissue will be a lot better to work on. The main problem with fresh tissue is that if you are doing such a long acquisition, your tissue will be changing over time, and we want to put into this fitting data which is in the same state, and that is very difficult to accomplish with fresh tissue.
Okay, thank you.
Maybe just to follow up on Hiramasa's question, when you do the postmortem imaging in the mouse, then do you do staining afterwards? Is there histology? And if so, what do we know about the axon sizes and diameter in the mouse compared to the human brain, basically? How much can we extrapolate given that the brain size is so different?
Very good question. So first of all, these were data from Mark Doss, so not my own. I haven't seen them with my own eyes in reality. The diameters, so to the last part of the questions, are of course different. So I think so we have some human data as well where we did segmentation, and we saw really that the axon radii are much larger. But since we have here both, we have the MRI data and the histology from the same subject available. I think at the moment it's the best we can get at. So what basically Jose did in his simulations, we have in the mouse model available. So G ratio changes, the radii changes, there are relative contribution changes. So in that sense, it's the most realistic available data set in my eyes, with all the limitations that histology has.
Just to follow up on the first part of the question, because I don't think we mentioned it, I believe it was transmission electron microscopy that was done on that data set.
Okay, sorry.
Just a bit more details for Kalanit. So a lot of this work is done with fixation with glutaraldehyde, which does shrink and probably non-linearly disproportionately affects myelin and axons. So I think, the talk about the elephants in the room, I would say that's the best one. And, Siawoosh shows a way forward. This calibration is important. There's other effects, vendor-driven effects, that will give you different results across sites. So I think this is really the way forward. Accuracy and the calibration is the only way to make G ratio work, not because the G ratio formalism is wrong, but because what you plug in there is too many things that are site-dependent, fixation-dependent, calibration-dependent.
So there's a question about what about in vivo studies when you cannot obtain ground truth values of G ratio for calibration? Any suggestions? I think maybe the way that it has been done in the past, I guess also by Nikos Stikov, is to look at, for example, the corpus callosum, where you know how G ratio varies or what kind of G ratio variations you are expecting to have across the corpus callosum, and see if you're in the right ballpark and if it is varying the way you expect. There was another question about how can you solve the problem of rotation in respect to B0? Can it only be applied to ex vivo or phantoms? Well, I come from the field of QSM, and in QSM we have been rotating heads for a long time, so we know how to rotate heads. But it's definitely not ideal. I think that we can start. So I showed two projects that one where we were using the rotations, the other one where we're doing multi-compartment relaxometry, where we were trying to help our fitting using the fact that myelin water has a very different T1 from free water, and I think mixing the two actually might work. So in one project we use the orientations, but we could also maybe use the T1 as a way to resolve these problems and integrate over the fiber tract, because there is no fiber tract that is always parallel to the magnetic field apart from the probably the cortical spinal tracts, but even those ones they bend a bit. Question?
Okay, so my question is related with the original benefits of G ratio mapping over just myelin volume fraction imaging or some myelin sensitive imaging. So I think Thomas showed one example of basically if you want conduction velocity, but presumably in most cases we just want to say some change in myelin, and then we get to this change of whether axonal volume fraction is biasing our myelin volume fraction estimate. But in the simulations you show, in the example you showed, you really needed to remove a lot of axons to see a subtle change, to correspond to a subtle change in the myelin thickness. So in practice, will the G ratio really give us a lot more sensitivity to demyelination compared with just myelin, imaging myelin, some myelin sensitive?
In a way, the more specific you try to be, the less sensitive you are.
Yes.
So I think we could make a scale of just T1 mapping, myelin water imaging, and G ratio. And so when we're trying to do the G ratio, probably we have the most complex model. It might be very specific, but the sensitivity will be very poor because you're fitting so many things. Myelin water imaging, I think it's at the state where we can actually have quite robust values, and it might be slightly more specific than T1 mapping. But if you want sensitivity, then just use maybe T1 mapping or MT. Like the most simple measures will give you the highest sensitivity They might not be very specific, but if it's just sensitivity you want to a myelin change.
Yeah, but I'm a bit worried that the specificity doesn't give you a lot of extra in this case, because you need a lot of axonal loss to kind of be an equivalent myelin loss.
I think it depends on the question you have. So sometimes, for example, you might really, for example, myelin might also change with when you're bigger, when you're male or female, so with volume, right? Whereas the G ratio is corrected for the volumes. So there are cases where that gives you extra information. It's also
Thank you.
Questions?
So sorry, we are over time. Those who want to go, please don't just go, and the rest can stay, and we can talk further.
So just to follow up on the last comment, and I agree completely that if your goal is to assess demyelinate or loss of myelin, then just a myelin-specific measurement would probably give you more sensitivity. But sometimes you would want to for example, assess remyelination and try to distinguish it from other processes such as resolution of edema in the case of MS. And so having better specificity would give you additional information. And also, in the MS context, again, you can't remyelinate axons that are not present. So having a measure that's calibrated for the amount of remaining axons that are available to remyelinate would be of use. So a measure of relative myelin thickness would be, I think, a helpful marker to have in multiple sclerosis if we can solve the issues of calibration.
So I would like to give a short comment on the question with in vivo data for those who are interested. So I think what we need at the end is something like we have at the moment for open source data available. So we need basically also open source histology and MRI, so that people for different scanners, for different sequences, and then we would be able to correct that actually, or other people would be able to correct that for their specific system as well. There's another question, yeah?
Thank you for really nice talks. I was just wondering how you can scale these metrics for different age groups, or if that's something that you're thinking about doing. Because I know that, I mean, for across lifespan or across development, and when there's like really early on when there's really no myelin, and then later on when that myelin actually develops, how do these metrics actually scale?
So I'm definitely not an expert on the exact biology that's going on, but it technically, what we're doing should be able to measure that whole myelination process early in development. There are always some methodological issues, such as are myelin metrics sensitive to axonal volume when you're completely demyelinated, that are, I think, open questions. But we should be able to follow this trajectory. There's been work by Dean and Dioni on looking at the developmental trajectory of the G ratio, for example, and it's a nice application of it, in fact.
Hi, and thanks for the talks. So I'm quite naive to this G ratio field, but I was wondering, there are lots of ratios that you are computing, and I was wondering basically the sampling space of the after these ratios, like they live in a constrained vector space, basically. Is it some concern when you are computing like scaling factors and so forth, or are there any like thoughts about it? Like for instance, when you measure a T2 star quantity, it is between 0 and infinity, right? But if you take measurements of two things, they would live in between 0 and 1, like the diameter, long diameter divided by, like short diameter divided by long diameter. And I was just wondering whether this, I cannot see anything. I was just wondering whether this like mathematical sampling space has any impact on the scalings and other stuff that we are computing. Yeah, maybe not.
It's a good question.
Well, I guess, for example, when we're doing like the deep learning methods, obviously you define the space where you can fit, and then that's where you can fit. You can fit anywhere else. And when you're doing these ratios, also you kind of make the noise not very homogeneous throughout the space. So you do have regions where you're more noise sensitive than other regions. And those would be my two comments. But,
yeah,
but we often need let's say ratios or some kind of ratio is very important in MRI to get quantitative results, because the imaging is not as arbitrary units. So also when you're doing relaxation metrics, in a way it is a ratio that you're doing.
Yeah, sure, but I think I will think about it further. Thanks.
Something else came to mind when you asked the question, though, because we are taking ratios, we do have to be careful if the fiber volume fraction is dropping to a very low value, because then you're just looking at 0/0 and you could get anything. As for the fact the G ratio is capped at 1, really, yeah, that probably has ramifications that none of us are commenting on explicitly at this time.
Yeah, I think thank you for staying so long with us, and we close the session with this. Thank you.
The most important question was, what was the letter type?
Good to see you.
Did you ask for the letter type? Sorry? Or was it you asking for the letter type?
For the what?
For the letter, the font type.
Oh, no, but I was curious, actually. That's a good one.
It's a Glasgow.
It's a Glasgow font?
Yeah.
Cool.
I didn't know that.
So have you seen these
Good to see you here. I talked about your work and I didn't realize you were in the audience at the time. Yeah, no, it's fascinating work. Yeah, sorry, I put the whole butt in about the head rotation. I mean, head rotation is getting popular.
Yes, yeah. I'm still hoping to get around it.
Oh, yeah.
Not necessary.
Yeah.
Should in principle be possible. Estimate and get a reference of the extractional phase, which should be somehow possible to get another way.
I don't know.
So basically, yeah. So I'm basically
we can replace it in the Malian water imaging formally.
Do my measurement of multiple D values, so I've got a different amount of contamination on my taxonal space, and compare that, and that should allow me to estimate both my taxonal and taxonal phase.
Excellent.
But for now, even